Okay, everyone, welcome to our biology seminar. It's my great pleasure to be able to introduce new faculty at Lees McRae, who's graciously agreed to come and talk to us about her research program. And this is Sinjani Goswami. She just started at Lees McRae this year and got her PhD at the University of Miami in Ohio in 2017. And she worked with Dr. Melanie Fisk, who used to be a professor here at App. So we have this cool connection. Before that, she had her master's degree at the University of Boku, perhaps that's how it's pronounced, with a joint connection with Austria and Finland. And before that, she got her undergraduate degree at the University of Calcutta. So she is an ecosystem ecologist and is hoping to set up some research locally. So anybody who's got an interest in ecosystem ecology and forest nutrient cycling things, definitely reach out so that we can make some personal connections eventually rather than just Zoom connections. Before I let her share screen and get started, are there any announcements for the biology department? No announcements today. Okay, well, thank you very much for coming today, and I'll let you share screen and tell us about your research program.
Thank you. I think, can you all see my screen?
Yes.
Okay, thank you. So first of all, thank you very much for inviting me to give the seminar. And I am really hoping to get some professional collaborations as well as personal collaborations out here today. I moved in from southwestern part of Ohio to northwestern North Carolina very recently, so I'm very brand new here, and all of the research that I'll be talking today is primarily going to be based off of my dissertation research that I conducted in northeastern part of the United States, so primarily around New Hampshire, northern hardwood ecosystems. So kind of giving you a little brief background of why nutrients and why really essentially, you can skip the bio slide, sorry. I wasn't sure if Lynn was going to introduce me or not, but so kind of jumping into the ideas to why nutrient limitation or even thinking about nutrients is so important, because they are such an important and valuable resource of any kind of plant growth, which directly, because plants are the primary producers, of course, that directly deals with the ecosystem productivity as a whole. And a traditional view of nutrient limitation has been coming from the Liebig's leaky bucket theory, law of minimum, which kind of indicates to us that the shorter the stav or the height of the stav, that particular resource is going to limit how much amount of volume of water you can hold in this barrel, which is the analogy of how much growth this ecosystem is going to show. So it's kind of limited by this shortest stav. And that's the more traditional view of ecosystem productivity being nutrient limited. But another thing to kind of keep in mind is, depending on ecosystem development, depending on whether the ecosystem is geologically on an older soil or a younger soil, the nutrient availability, especially of the two primary macronutrients that are nitrogen and phosphorus, depending on the Walker-Sayers model, 1976, we know that ecosystems that are primarily in younger soils or developed off of younger soils, they're primarily nitrogen limited because they're still accumulating that amount of nitrogen through the system. Whereas geologically older soils or ecosystems developing directly from geologically older soils are going to be more prominently phosphorus limited, because phosphorus over time is gradually going down in the soil, not only just because of fixation, but also being utilized by the ecosystem productivity, and over time for phosphorus in the system gradually goes down. So these are kind of traditionally thinking about nutrient limitation that kind of should indicate that northern hardwood forests, which are primarily geologically younger soils, and I'm talking about central New Hampshire, I'm talking about White Mountain National Forest, forests, forest ecosystems that are very close in terms of the species dominance and characteristics that we find here, but geologically those soils are relatively much more younger. And so having such a relatively younger soil would indicate that probably nitrogen and not phosphorus would be the likely limiting nutrient in such kind of ecosystems. But on the other hand, if we think about resource optimization theory, we know that vegetation kind of changes or adapts to which type of resources are available around it, and more primarily it can shift its allocation patterns depending on, is nitrogen more available, is it phosphorus that is more available? If both of the nutrients are available, shifting allocation below ground to above ground, these are various ways that primarily vegetation can shift. And the more ecosystem ecologists have kind of moved away from thinking in a traditional single nutrient limitation ideas to kind of more like multiple resource limitation or multiple nutrient limitations based on these resource optimization ideas that things can shift over time, even within the context of the same ecosystem. And a very relatively recent synthesis of several nutrient fertilization studies across a board of or a host of different types of ecosystems, say for example, terrestrial, freshwater, marine, Elsewhere's study, a meta-analysis of several synthesis of fertilization studies indicate that when you add both of these nutrients together, there's a significantly higher response of or productivity response in these different types of ecosystems. But interestingly, off the terrestrial bar that you see, basically with the purple bar where N and P are both added, that is significantly higher where my cursor is right now, significantly higher than if you add nitrogen alone or if you add phosphorus alone to the system. N and P added together elicits a big higher response. And that is across the board with terrestrial ecosystems. But interestingly, out of the 100 studies that was pulled by Elsewhere's work, only 4 of them were temperate forests. And so temperate forests are a type of an ecosystem that doesn't have a very high representation when it comes to figuring out all of these nutrient dynamics and how do we shift from that traditional view of nitrogen limitation, nitrogen limits, temperate forests to exactly understanding what different types of mechanisms are operating under it, and has it shifted based off of resource optimization. So kind of giving you an idea of where all of these study sites are located. How do I move this Zoom bar? I'm sorry, I'm just trying to, yeah, that was, yeah. So if you look at this area of central New Hampshire, traditionally this area has a legacy of nitrogen deposition because of being proximate to locations like Boston, Connecticut, and so on and so forth, which has quite a high rate of nitrogen deposition. So yes, of course, right now nitrogen deposition levels are, this is a figure that is 2 years, 3 years back dated, but what's important to keep in mind that even if nitrogen deposition rates are going down, but these areas, these forest ecosystems could still have a legacy of high nitrogen deposition since the 60s, 50s, and so on and so forth. So that kind of brings me to that question, such a strong and long-standing legacy, excuse me, of nitrogen deposition, could it elicit or induce some sort of a phosphorus limitation in these ecosystems when we think about ecosystem productivity? And based off of not ecosystem level experimental studies, but based off of ecosystem level modeling results done by Ed Rastetter and resource allocation ideas, it kind of suggests that multiple element limitation is becoming more and more common in various different types of terrestrial ecosystems, and especially temperate forests, which if you separate them by a successional stage of development, relatively younger forests that are somewhere between 20 to 30 years old, right after clear cut, they kind of show more phosphorus limitation. And gradually, as forests are getting older, we're moving towards 100 to 200 years old after clear cut. Those types of forests are kind of supposed to show both nitrogen and phosphorus co-limitation based off these modeling studies. So that kind of prompted me to understand, can I do an ecosystem level study to kind of figure this out? And how do I detect it, first of all? So like, ideally thinking about co-limitation, the word, what that basically means, co-limitation by nitrogen and phosphorus, basically means when you add both of them together. So say, for example, if you think about the N plus P together, that is supposed to be, if a system is co-limited by both of these nutrients, you're supposed to have a higher response when you add them together than what you see as individual responses. And that could be an additive format of co-limitation. So what that basically means is you have a significantly higher bar for N and P. If you basically add up these individual responses, this bar is going to be higher than that, or could be equal to this plus this individual responses. So that could indicate a format of co-limitation. The other format of co-limitation is having a synergistic effect or some sort of an interaction between both of these macronutrients, so N and P. And you are probably going to see a huge effect, a much greater interaction, which is different than if you individually add these stuff together. And this response to N plus P added together is going to be significantly very different when you compare it back to individual additions of those nutrients. So these are two ways to kind of detect co-limitation. And the interesting part of an underlying mechanism of additive co-limitation is it can follow up in a sequence. Say, for example, in one case, you could barely have any response when you add nitrogen to the system, but you get a higher response in case of a P-limited ecosystem. When you add phosphorus, you get this red bar is pretty higher relative to the control. But when you add nitrogen after you have alleviated the P limitation, and you see that response showing up when N plus P is added together. So that's almost like a sequence pattern. One happens after the other one. So basically, it could happen with, on both sides, both ends. It could be a P limitation, initial P limitation followed by nitrogen limitation, and that causes the N plus P bar to go up, or it could be an initial N limitation followed by a P limitation, which causes this N plus P bar to be incredibly higher. So these are kind of ways to detect co-limitation in a system. And so that kind of brought me, I kind of put my entire dissertation within those basic frameworks of question and kind of wanted to test them at not only at an ecosystem level, which is the first question, is above ground productivity or forest productivity co-limited by nitrogen and phosphorus in the northern hardwoods, or I kind of wanted to tease apart certain allocation patterns. And well, for obvious reasons, it's not possible to cut out big, ginormous ecosystem trees and understand allocation, but I kind of wanted to know what's happening in the forest floor as well, and kind of took seedlings that are representative of the main major dominant plant species of these ecosystems, and I kind of wanted to notice what their survival and allocation patterns are going to show up me with respect to as a response to nutrient additions, the same nutrients being added at the same rate and so on and so forth. And not only that, I wanted to dig a little bit deeper and go into the below ground and understand some soil responses and do direct interactions between soil and nitrogen availability and phosphorus availability. Do they interact in the soil to kind of show up as patterns above ground and so on and so forth. And these are various ways that I wanted to detect co-limitation. And the very first kind of thing, telling you about a little bit about this particular project that I was putting all my dissertation under, big umbrella of this project. This is called the multiple element limitation in northern hardwood ecosystem. It's a big project that kind of is all together in central New Hampshire, White Mountain National Forest, so kind of located right here. And it's spread across on 13 different stands that are situated within 3 separate sites. So the 3 separate sites would be Hubbard Brook Experimental Forest, Jeffers Brook, and Bartlett Experimental Forest. And within each of these sites, as you can see, there are a whole bunch of these black dots that are nested. This is what I'm calling as stands. And these 4 stands, a total of 13 stands, and each of these 4 stands are about 50 meters by 50 meters. So it's a quite ginormous plots of 50 meters by 50 meters in each of these 4 stands. And there's an age gradation of these 4 stands. So there are several forest stands that are about 25 years old after clear cut. There are several that are 30 to 40 years. That's what we are calling as mid-age. And then there are several that are mature, which are greater than 100 years in age. And in each of these 4 stands, or each of these 13 stands, you have 4 separate plots or treatment plots. And one of them, these are 50 by 50 meters. Sorry, I talked about 50 by 50 meter ahead of time, but each of these little squares is what is of that scale. And if you look at this map, what's happening is you leave one 50 by 50 meter as a control plot, and then the other one you add just phosphorus to it. That's represented as the red color, just nitrogen, which is represented with the blue color, and the N plus P together at the same rate as it was applied individually, in this represented as the purple color for a stand. So please remember the same color pattern is going to show up later in the results as well. So to kind of give you an idea of what rate of application of these fertilizers are happening, nitrogen we were adding since 2011 at a rate of 30 kilograms per hectare per year. This is an annual addition that's happening since the start of the fertilization study. And we are adding them as the format of ammonium nitrate pellets, and which is kind of directly giving you a lot of ammonium and nitrate in the soil directly for picking up. And on the phosphorus end of things, we are adding about 10 kilograms phosphorus per hectare per year as sodium monophosphate powder. And just to let you know that the difference in the rates are not stoichiometrically balanced. The nitrogen rate is almost about half of the annual net nitrogen mineralization in these ecosystems. And phosphorus, we're adding a little bit more of phosphorus than the annual rates of P mineralization, because there's a propensity of phosphorus getting stuck in the soil or getting fixed in the soil because of pH changes and so on and so forth. So that's kind of the background of how much fertilization is happening since 2011. And to kind of achieve the goal of that first question of understanding if above ground productivity is co-limited, I asked certain specific research questions. So is tree growth at an ecosystem level co-limited by both of these, or does a single nutrient limitation still prevail in the other ecosystems? And does the limiting nutrient have anything to do with which type of age this forest is in? So is it younger, older, or mature, and do they differ of which type of nutrient they're limited by? So kind of telling you how I collected the data, we measured trees that are greater than 10 centimeters in diameter, and they were measured prior to start of treatment and since 5 years after treatment. And the variable that I measured was relative growth rate. And we estimated that based off of per tree relative basal area increment. And just to give you an idea of how I did the stats, I basically divided the nitrogen treatment as control and nitrogen treatment versus no nitrogen treatment, and phosphorus treatment versus no phosphorus treatment. And if you look at what is coded under nitrogen treatment is both plus N and N plus P, and no nitrogen would be control on phosphorus. And in cases of phosphorus, you have plus B and N plus B. So anytime you're adding that element in that combination, you're considering that as that added treatment. So to kind of first see what we found was a couple of things about this figure before you start to notice it. So you'll see that in the x-axis, we have young, mid-age, and mature forest stands. And on an average, you'll see that the relative growth rate of increase in per tree basal area for these stands, the younger forests are kind of about 7.2 and above % growth per year. And the mid-age forests are growing around 4.9 to 5% per year. And the mature forests have a much lower growth rate for obvious reasons. And they are growing about 2% per year. And within each of these 13 stands, we found that there's if I clearly show you the control nitrogen, phosphorus, and N plus P, you will start to notice that the purple averages are slightly higher when compared to the control if you look at both mid-age and mature. So out of the 13 total stands, in 6 of the stands, we found a clear, obvious higher high growth rate after N and P were added together. So, and then I kind of wanted to tease that apart. Like, is it just N plus P together, or is it no N versus no P, or no N versus N, and so on and so forth. So if you look at the next figure, which is here, the relative growth rate, if you look at the mid-age and the mature forest, anytime phosphorus was added, it had a significant higher growth response. In terms of mature as well, so the most clear prominent effect was in the mid-age, slightly higher effect was shown in the mature, and no effect was shown with P addition in the younger forests at all. And with nitrogen addition, it was noticed there's a suggestion of a little bit of a nitrogen limitation suggestion, or a higher growth related to the no nitrogen additions in the younger forests, but this wasn't a significant pattern, so we can't really be very confident of this. And nitrogen did not elicit any response to nitrogen addition in both mid-age or mature forests. And we did not find a significant N by P interaction, which kind of canceled out that co-limitation idea. So there was no NNP interaction found in any of these ages. It was a straight up P limitation effect that we were seeing. So conclude that part of the ecosystem level response, forest productivity at a ecosystem scale in northern hardwoods, especially in mid and mature stands, it's still single nutrient limitation, and that is P. A phosphorus, add phosphorus from outside, it shows a significant high amount of productivity response. And responses to phosphorus, even though they were relatively modest, but I think what's going to happen is this is 5 years after treatment beginning. And so the more time you give, these responses are going to shift over time. And it's important that we kind of follow up with what's happening since then. But these could also be a consequence of, say, differences in the age, differences in the spatial heterogeneity of where these stands are located. And that could kind of interfere with a straight up response to phosphorus. And interestingly, there are several, the convincing part of it was out of 13 stands, this is a result out of 13 stands, which is it's a substantial number of stands that is showing you this particular result. So that's all automatically almost like an ecosystem productivity result. So that was the cool part of this particular project. And even though I was gung-ho about co-limitation, I couldn't find any clear evidence of co-limitation at an ecosystem level. And to kind of answer that why phosphorus limitation, it's not expected based off of that geological, like Walker-Sayers model, and geologically these are younger soils. They shouldn't be phosphorus limited, but like I say, these types of ecosystems have had a very strong amount of nitrogen deposited over time into these ecosystems, and that could kind of, lead up to a situation where phosphorus limitation is almost acting as an induction after relieving or having a legacy of nitrogen being deposited in these ecosystems for a high rate for a very long period of time. So that's kind of the way that I interpret these results. So kind of that was the ecosystem level study that I conducted. And then to go in a little bit more about the understory, the forest floor, and where more allocation mechanisms could be talked about. And I, first of all, many people say, oh, seedlings are not same as big trees. Of course, they're not same. I'm not nowhere saying that they're going to have the same response, but it's important to kind of think about the same species, what exactly happens with the seedlings, even in that context of adding nutrients at the same rate. So I conducted in the same exact stands, just I chose a subset of those stands, and I wanted to see what is happening with these little seedlings of sugar maple and beech. These are codominant species up there. And so the first thing to think about whenever we are thinking about these little seedlings in the forest floor is they do have a possibility to be nutrient limited, especially in highly shaded conditions. So they're always under a deep cover shade. And so if nutrients are being added from the outside, is that giving any of these species some sort of an advantage in terms of their survival? Or do they directly shift their allocation below and above ground in response to these nutrient additions? So if we do end up alleviating nutrient limitation, ideally, that should directly result into some sort of a shift to above ground. So if they do have plenty of nutrients available in the soil, they're not going to explore a higher degree of soil volume to get those nutrients. So for example, I thought that the best way to kind of test allocation in this case would be to measure directly whether these little seedlings are shifting their allocation above versus below ground or not. And so that could happen in two ways. They could directly take up and put in a lot of above ground biomass, or they could grow above ground more, or at the same time, they could decrease the amount of roots that they're making, and did that almost like a mediation effect, add that carbon back up. And so but that kind of comes also, shifting of allocation also comes with a baggage of some incremental costs. Say, for example, if you add up a lot of carbon above ground, you could kind of potentially put the individual seedling at a risk of increased herbivory damage. So it could be attractive if there's more nutrients itself in the tissues, as well as not just carbon, but a lot of nutrients, that could become a lot more palatable of a food source to several different herbivores. And so that itself could again loop up here, like it could increase, like adding nutrients from outside could increase herbivory damage, and that could lower above ground growth as well. So that kind of mechanistic conceptual model is what I based off my next set of research questions on. So there are several allocation efforts that could be measured. Say, for example, biomass. That's one of the first things that show growth. Is it growing up more or not? Is it heavier or not? Is it investing a lot of carbon in it or not? So biomass is something that potentially, once, if there is no nutrient or nutrient limitation is alleviated, biomass should go up right away. So the whole seedling biomass. So biomass is an allocation variable that could easily show, my prediction is that should go up. And SRL and SLA, these are specific root length and specific leaf area. So that's talking about carbon efficiency or carbon use efficiency more. So if you're alleviating the nutrient limitation, that should you should invest less amount of roots per unit of carbon. So specific root length should increase. specific leaf area, how much leaf area or leaf tissue are you investing for the same amount of carbon that you use. So specific root length, specific leaf area going up is increasing carbon use efficiency. And so adding nutrients from outside should cause that to go up. And for obvious reasons, the foliar, the tissue should become more nutritive. So that N2P concentration, if nitrogen is added from outside, that should go up. So foliar nitrogen should go up. And at the same time, herbivory damage could be a potential way to think of it. So leaf area damage might go up as well because they're becoming more nutritive. And on the other hand, variables like root to shoot should go down, or that is the ratio between how much roots you're making versus how much shoots you're making. So you should make less roots and you should make more shoots if nutrients are added from outside. And similarly, foliar C to N, which is the carbon to nitrogen ratio or carbon to phosphorus ratio, those should go down right after nutrient addition. And so all of these different types of seedling allocation efforts could indicate some sort of a response to elevated nutrient availability. And kind of my main three questions under that was, is seedling survival nutrient limited? So if I'm adding nutrients from outside, is survival going to be going up for both of these dominant species or not? And secondly, biomass, is it going up or down? And biomass, is it directly related to all of these allocation patterns that I was talking about, or to root and shoot foliar morphology? So in order to kind of test that, I chose 3 out of the total 13 stands. And out of those 3 major big mature stands, I went and demarcated 10 seedling subplots that are 1 by 1 meter. And within each of these 1 by 1 meter, as you can see here, I measured various different like survival. I went and tagged the survivors. And one interesting thing, there was a mast year in the year 2011 when we started fertilization. So we had a ton of seedlings crop up in the spring of 2012, which was literally right after we began fertilization. So I utilized that mast year of sugar maple and beech to kind of start this seedling study. And all of the survivors, or all of the first year ones that were germinants, that I called germinants at that time, that were the direct result of that mast year, I went and tagged them with zip ties, so that I can come back and look at survival and how many of them were alive over time, over years or not. And not only that, I also went to the outer buffer area, so not directly in the 1 by 1 meter plots, but outside of these each of these plots, and I destructively harvested a whole bunch of the same age beech and sugar maple, and I measured the age basically based off of the scars, the bud scars of these seedlings, and I destructively harvested those, and I brought back to the lab to kind of measure a whole bunch of these allocation efforts. And as you can see, these are some examples that I wanted to do the root length. I wanted to do how this is a beech sugar maple, and we measured the leaf area using ImageJ. We measured the root length, we measured the masses, and so on and so forth. So allocation efforts kept me very busy for several years. But interestingly, the couple of important results that I want to share with you are, the very first thing, survival, for obvious reasons, very few seedlings survive over time. And for both of these species, seedling survival showed a drastic decline over time across all of these stains. And so, as you can see, we once upon a time started with about more than 5,000 seedlings. And over time, at the end of each cycle, after 3 years or so, we basically were down to about less than 500, or somewhere between in each of these stands. So yes, survival will get hit, and we know, because not everything that's germinating is going to be able to survive. And interestingly, what happened is, when you go look at the survival patterns of both beech, the beech is panel and the top, panel A, and sugar maples in panel B. In each of these stands, the thing to notice here is between these treatments, when you add nitrogen, which is, with circles, black circles, you will start to notice that all of these black circles are towards the very lower end of survival. So that, especially in beech, which is one of the most important dominant species up there, there's a strong suppression of seedling survival related to the control. And that pattern kind of stood up for both all three stands. And in sugar maple, what was interesting was over time, in sugar maple survival kind of declined with whether you added nitrogen or phosphorus, irrespective of, or whether you added them all together. And so in both of these dominant species, seedling survival showed a direct decrease after nutrient additions. And to kind of go back and talk about why is survivorship declining? Does it have any direct relation with any of the allocation efforts? Here's a little bit of specific root length, and it's showing you the exact same control versus nitrogen versus phosphorus versus N and P together. And even though the pattern is not prominent in any way for beech, but if you look at sugar maple, when you add N and P, kind of especially the nitrogen addition. So if you look at C8, C9, kind of lower, but C8 and 2 out of 3 stands, you always see that nitrogen addition has a much higher specific root length than control. And those N and P added together also has a higher pattern related to the control. So that kind of indicated to me that something's going on, and we need to kind of dig in a little bit deeper. And they are shifting their allocation in certain ways, but it's not uniform across the board. And unfortunately, I didn't find seedling biomass or root to shoot or specific leaf area to change. They were relatively unresponsive to nutrient additions, but the only allocation effort that I really clearly saw was this specific root length change in sugar maple. And interestingly, if I looked at the nutrition part, like whether their tissues are becoming, whether their foliar tissues, the 2 leaves or 3 leaves up top, if that is becoming more nutritious or not, you'll start to see, I know there's a lot of box plots here, but the interesting thing to kind of notice is over time. So year 3 and year 3 for both. The top panel is foliar C2N for beech. This is beech. This is C2N for C2N for sugar. Sorry, please ignore this sugar maple. I don't know why I put that. This is foliar C2N of beech. This is sugar maple. This panel C is foliar C2P of beech, and I forgot to add the sugar maple here. So if you look at both beech and sugar maple, you'll see that their nitrogen content is increasing in their tissues, and their phosphorus content is increasing in their tissues with nutrient additions. So both foliar N and foliar P were higher where these nutrients were added. So they're over time, relative to control, they're becoming much more richer in nitrogen and phosphorus content. And interestingly, what happened is the leaf area damage added up. And so if you look at beech leaf area damage, which is panel A, and sugar maple leaf area damage, which is panel B, you will start to notice that relative to the control, which are all these white bars, the nutrient added leaf area damage, which herbivory damage would look like this. This is an example of a beech seedling showing pretty high amount of herbivory damage, both in beech and sugar maple. The damage was much greater related to the control, and that was significantly higher, especially in beech. Wherever you added nitrogen, whether in nitrogen alone or whether in a capacity with nitrogen and phosphorus, those were significantly higher than control in terms of leaf area damage. So Sugar maple leaf area damage, even though it showed such a high rate of increase, but since it wasn't there for all of the, there was a huge amount of variation, as you can see from these error bars. It did not directly significantly respond to NNP addition. However, that pattern was pretty clear for beech. And interestingly, I wanted to see if survival had a direct correlation with the leaf area damage that I was seeing. And survival was not affected, interestingly, but there was a very, very weak trend in sugar maple with survival kind of going down with increasing leaf area damage. So there was a whole host of pretty interesting things that stood out to me with this particular study. Seedling survival was directly suppressed by elevated nutrient availability. And I also noticed that there was a lack of plasticity in seedlings. Improved nutrition had little effect on growth allocation. Biomass did not change. Root to shoot ratio didn't show any change, which I was thinking they should be even more plastic, but I did not find such plastic effect on of nutrient additions on growth or allocation. So, and the way I explain the nitrogen suppression effects is it's probably an indirect effect of acting through the species susceptibility for stress or drought or determinants like, say, physical damage or pathogens. And very interestingly, which I want to pursue further, is actually if I followed up for more than 3 years, how is this pattern going to change? There are some survivors, and they're very limited in number, the ones that are still living in those plots that are less than probably 20 or less in each of the plots, each of these 30 by 30 meter plots. It would be interesting to kind of follow up with it, but given such a drastic lowering of numbers, it was difficult for me to continue on with that. So that was kind of, I feel like I want to restart a study like this somewhat closer here and kind of figure that out with dominant species. And kind of trying to go, I know I'm going to probably rush through the last few slides, but kind of digging a little bit deeper under the soil and figuring out if soil nutrients directly interact to contribute to NNP co-limitation. I kind of made this conceptual model that will help me to understand that if you add, of course, this is just an ecosystem where you see that there's various different types of processes that are connecting the available nitrogen and phosphorus in the soil solution. So that's indicated by this box, which says soil solution inorganic nitrogen and phosphorus available. But that is directly a consequence of how much mineralization and how much microbial uptake is going on in the soil. And so the soil organic matter and the microbes is kind of in their own box because they are limited by nitrogen and phosphorus availability as well. So I'm kind of thinking in terms of an ecosystem level, there could be various interactions that are happening between these two steps of mineralization and microbial uptake that could change the size of this box, like how much nitrogen and inorganic nitrogen and phosphorus is available for plant uptake. So could these internal below ground mechanisms or interactions, could they have an effect to showing what kind of responses we're seeing at an above ground level? So in order to kind of think through that conceptual model, what would make sense is if you add nitrogen from outside, if you add phosphorus from outside, there will be less amount of mineralization between your microbes converting organic matter into inorganic nutrients, because that amount of inorganic nutrients you're adding from outside. And at the same time, the extra added elevated nutrients that you just added in the inorganic form could include in microbes bumping up their activity and their respiration, their biomasses in their body. So microbial uptake could be increased or elevated directly as a consequence of adding inorganic nutrients from outside. So kind of trying to understand that whether that would have an internal effect on above ground productivity. I wanted to know what is the effect of one nutrient added. Could that, an excess of N change the availability of P, or an excess of P change the availability of N in the soil itself. And in order to figure that out, I used resin strips. And these are pretty nifty, cool ways of figuring out how much inorganic nutrients you have. These absorb the cations and anions in the soil, and you can extract them once you put the resin strips inside the soil and you let it incubate for 14 days. Then you go back and extract those strips. They've automatically absorbed a whole bunch of those inorganic cations. And once you extract them, you can bring back to the lab and analyze how much nitrate or ammonium or all of the phosphate is available in the soil. And this is again inorganic in the available nitrate, ammonium, and phosphate that are in the top 1 to 10 centimeters in the soil solution, basically, not further down. So not only those resin strips, so I in order to find nutrients in the soil solution, I used resin available NNP, but I also simultaneously did a whole bunch of microbial respiration incubations, so carbon mineralization that kind of gave you an idea of how much these microbes are chomping up a whole bunch of NNP. And I also did a whole bunch of net nitrogen mineralization lab incubations, brought the soil back, and then kind of did the incubation to figure out what was the main source of nitrogen to the soil solution. And so these are just some lab images, and also did an extraction of bicarbonate available phosphorus to kind of indicate how much is the potential supply of the soil of phosphorus in the soil solution. So did a whole bunch of these variables, and the very interesting part was, if you look at these figures, this is since fertilization, 2 years, 4 years, 6 years. That's collecting resin strips, or basically using resin strips each year, and figuring out if addition or treatments of N plus P together, or phosphorus alone, and control is depicted here in yellow, does resin available inorganic N change over time? And interestingly, if you look at relative to the yellow line, your red line is gradually going down over time. So that's indicating in the phosphorus treatment, there's a clear suppression of available inorganic nitrogen that is present over time, and that's gradually going down because of the phosphorus treatment. And obviously, when you add nitrogen to the system, it's going to increase the available inorganic nitrogen. And this was a very startling pattern. So there's a clear suppression of one nutrient because of the excess of another. However, that pattern I didn't find as a significant pattern across all these stands in phosphorus. So resin available phosphorus, even though it was slightly less, but it wasn't significant suppression of nitrogen addition on phosphorus availability. So the bottom panel is showing you resin available phosphorus over time in all of these treatments. So resin P was greater in P plots. P and N plus P had a higher resin P availability, but this blue line, I wanted it to go down, but we want a lot of things in science that doesn't immediately show up. But so that kind of told me that there's, again, something going on. And I wanted to look at the potentially available mineralization index, like how much is it available, not just in the soil solution, but potentially available nitrogen using net nitrogen mineralization incubations. And if you look at here, there are 2 lines is the red line is indicating P addition, and the blue line is indicating no P addition. And if you look at, and this is no N and N, if you look at nitrogen mineralization, this is the top panel is basically organic E. Then you have the next layer, organic A, in the middle, and then you have the mineral horizon, which is further deep down. And interestingly, the nitrogen mineralization was suppressed by N plus P addition in the mineral soil. That was the more like these top ones. The net nitrogen mineralization basically increased in response to nitrogen in organic soils. but decreased in response to phosphorus in the mineral soil. And that was kind of one of the really cool results that I wasn't expecting, but I was expecting this all through, but it didn't show up in the organic soil. So again, another addition of another nutrient is kind of altering the availability of the other one. And potentially available phosphorus using bicarbonate extractable phosphorus, it didn't really show much of an interesting pattern. It did increase in response to phosphorus in organic soil, but not in mineral soil. And kind of tying back together all of that story of based off of that conceptual model that I started with, that yeah, I kind of did see a little bit of an increase in microbial uptake of nitrogen and phosphorus, but I did not see the decrease in mineralization of NNP that I was expecting based off of these outside additions. And that kind of brings me, tying it back to the first, the above ground productivity response, that plants grew more or ecosystem showed a higher productivity with adding phosphorus in the system. So this whole thing that increasing plant uptake because of P addition, so having more phosphorus in the soil is causing more phosphorus to be taken up by the plants and growing more. That probably, in addition to these microbial uptake going up, but mineralization not going down, it's probably decreasing the soil solution or the available amount of nitrogen and phosphorus in the soil. That's what is possibly an effect of microbial uptake of nitrogen suppressing the amount of phosphorus that's available to plants. And so it's kind of interesting to tie all of these back together. So elevated phosphorus availability kind of suppressed nitrogen availability after 5 years of fertilization. We've been fertilizing this long, and microbial growth appeared to be phosphorus limited in these types of ecosystems. And so my new hypotheses to kind of move forward with this would be, would alleviating microbial phosphorus limitation induce nitrogen limitation over time? So I'm very gung-ho about teasing apart these individual nutrient additions and effects that are showing up over time, and mechanisms that we don't, it's happening in the black box, but we don't see it. So that could, over time, if these stands are allowed to be monitored with these same responses and get fertilized for 10 years or so, and more than 10 years, we could kind of start to see or notice a sequential co-limitation response over time, which yet is not showing up. So kind of summarizing all of these together, ecosystem productivity as of now is not NNP co-limited. We didn't see that significant interaction and a high response to N+P across all the stands. P limitation is something that we have already noticed and evidenced in mid-age and mature forests. Even though they're spatially variable, they're showing a pretty convincing pattern of P limitation as of now. And seedling growth, to kind of tie in a little bit of how allocation shifts. Seedling growth, I couldn't find it to be nutrient limited or co-limited, but improved nutrition might be, and was probably detrimental to seedling survival after nutrient additions were occurred in these ecosystems. And below ground responses to kind of suggest a little bit of microbial suppression of plant available nitrogen. And that kind of brings in to this closure of this whole study. And I want to kind of continue forward, leading and understanding how ecosystems over time change these interactive mechanisms to be single nutrient limited, co-limited, and so on and so forth. So with that, I'm at a close, and I have several different people, my ex-dissertation advisor, Melanie Fisk, and a whole bunch of scientists working up at Hubbard Brook, and several undergrads. And without several different grants, none of this work would have been possible. So that's kind of it. Thank you for an opportunity to share all this with you.
So do we have any questions from the audience?
I have a question. Good talk. I'm kind of a soils nerd by profession, but have you did you guys look at all at the cation exchange capacity of soils, or do any soil descriptions or anything to see if there was something limiting there, like they had low CECs?
Yeah, we did not measure cation exchange capacity directly, but since I was so gung-ho about phosphorus, I also wanted to pair up with a pH and calcium effects if there were any. So I did measure some calcium in the controls and calcium in rest of the other treatment plots, and I wanted to see if the phosphorus is getting fixed into calcium or if there are any, but nothing really stood out to me that I could, yeah. So their pH is pretty much close to about 4 point something in terms of average pH across these types of soils. But sorry, no, I did not measure cation exchange capacity, but I was very interested to know where the phosphorus was going. So hope somebody else will do it.
I had one other question to you, just about the site and the differences in the site. So, it sounds like, some of these forests were heavily timbered a long time ago and some more recently. Do you think that, some of the differences could be due to like the stripping of some of the soils at the surface? Basically kind of as they're logging these areas, I mean, they usually do a lot of damage to the soils.
True, but like I said, like all of these were experimental forests that were long since not being logged, but there were some that were relatively recently. That's where that the young forests come into play, and it could be a direct consequence of that, but I haven't yet explored any what would be the direct effect of logging into the soil damage. So I don't have a ready answer for that. Sorry.
Thank you.
No worries. Thank you.
I go ahead and ask a question?
Yeah.
Shinjini, good talk. Maybe you can help me with my biogeochemical cycling, because I seem to remember from when a friend of mine was looking at phosphorus limitation in pitcher plant bogs in southern Georgia. He made mention that underappreciated by a lot of people, but phosphorus can sometimes go off as a gas. So I was wondering if you knew anything about that, and that may be just another way of inducing a phosphorus limitation. And the other thing was, do you have any feel for changes due to mycorrhizae?
Yeah, thank you. So I was hoping somebody would bring up mycorrhizae. To answer your first question, there might be a little, not very well understood mechanism of phosphorus being lost as a gas. I think majority of attention goes to denitrification end of things from soil, but I personally haven't looked a lot on phosphorus losses as gas, but that could be a potential way. But I have zero experience with bogs, so that is something that I don't know about that. To answer your mycorrhizae question, so I was more interested because you saw I was doing seedlings that effectively beech is a ectomycorrhizal and sugar maple is a arbuscular mycorrhizal. And so a more recent follow-up study is going to happen with the above ground ecosystem productivity response. And it seems to me like when I was dealing with the data, ectomycorrhizal species totaled up together were showing a higher B response. So when you add phosphorus from outside, beech, white birch, all of these species were having a significant phosphorus response, a higher response to B addition. On the other hand, sugar maple, aspens, even though there are several, the big tooth aspen and all of them are they can form both types of mycorrhizae, but they were showing AM species combined together. These I'm talking about the big trees. They were showing a little bit high response to nitrogen addition. But however, this is what I'm talking about after 5 years of fertilization. So I've recently heard that Noah Blumenthal, who's currently Melanie's dissertation student, he is currently looking into the differences between the ectomycorrhizal and arbusculomycorrhizal. And to tell you a little bit more, Howie, for the seedlings, I actually, from the destructively harvested seedlings, I collected a whole bunch of root systems, and I wanted to do the mycorrhizal colonization of EM versus sugar versus beech, and but I couldn't complete that. And but I was man, that amount of dissertation work I've done in the past 5 years, it was a little too much. It chewed up a lot. But a little story is for the little seedlings, I was seeing a pattern of, again, pretty similar to the big tree response, the beech seedlings, where their root knot, the ectomycorrhizal colonization, the mantle, that was slightly higher with bee addition. But this is, again, not gone through statistical analysis. So there is some exploration that we need to do about the EM species directly with this bee addition. Is that helping them or not? Which is what is a good place to start for somebody. Yeah. Thank you for asking that question. I get pumped about mycorrhizae all the time.
Thank you.